Hello students and welcome back to the chapter, and in this module we shall be studying DNA library and its first part, genomic library. So let us begin with DNA library. So what is a DNA library? DNA library is nothing but the DNA of an organism that is stored and propagated in microorganisms. So here I have a human, and what do? We isolate its DNA and we put this DNA into a suitable microorganism. And what do? We allow this microorganism to undergo cloning or form multiple copies, and the collection of these multiple copies or the clone of the organism is nothing but the DNA library. So why is DNA library constructed? In nature, we see a lot of animals, or we basically see a lot of species. So DNA library is the collection of DNA fragments from a particular species. But what DNA fragments or which DNA fragments? Remember that they are specifically genes. So collection of DNA fragments that is specifically genes from a particular species is nothing but DNA library. And what is done with the DNA? They are stored and propagated in microorganisms. By which method? By the method of molecular cloning. So DNA library is the collection of DNA fragments from a particular species that is stored and propagated in microorganisms through the process of molecular cloning. Now let us study what are the types of DNA library. The first type we have here is genomic library, and we have cDNA library, and in this module we shall be dealing with genomic library. So here we have genomic library, and what is its definition? Genomic library is the collection of all clones of DNA fragments that represents the complete genome of an organism. So this is the definition of genomic library. Remember that, students. Now let us take a look at how a genomic library is constructed. So here we shall be studying construction of genomic library. Here I have a cell, and can you see the DNA in the cell? We isolate this DNA, and what do we do with it? We cut this DNA with the help of restriction endonucleases to get DNA fragments. Okay, and then we take vectors from suitable microorganisms. So here we have vectors. We also cut these vectors with the help of restriction endonucleases, and we combine these vectors with the DNA fragments to get a recombinant DNA or rDNA. And these recombinant vectors or rDNA will now be inserted into the microorganisms for multiplication or cloning. So here we inserted the rDNA into microorganisms. And what do? We allow these microorganisms or these cells to undergo multiplication to get a number of copies. So here they are forming clones, or they are forming a number of copies of the genome that they are harboring into the recombinant vector. And each of these copies will be stored separately. So that is a genomic library. Now let us take a look at the properties of genomic library. Now, when we cut the DNA or when we fragment the genomic DNA, we cannot know that which DNA fragment contains the gene, and hence we clone or we insert all the DNA fragments into the vectors for cloning. So we do not left out any DNA fragment. So when genomic DNA is fragmented, it is not known which fragment has the desired gene. Hence all fragments are cloned and the copies are stored separately. But how do we know that which fragment is actually having the gene? We can check that, or we can screen for these genes with the help of complementation or probes. Okay, so these are the properties or the functioning of genomic library. And in this module we studied DNA library and we studied genomic library. and in the next module we shall be studying DNA library and the second type, that is cDNA library. So till then, take care, study well, and bye bye.